So here's something kind of wonderful to think about as you're lying there, settling in for the night. You are, right now, in this very moment, preparing to do something that every human being who has ever lived has also done. Every emperor, every peasant, every astronaut floating in the International Space Station, every baby born this morning in a hospital somewhere, all of them sleep. And yet, for most of human history, nobody had the faintest idea why. Not really. Not in any way that would satisfy you if you actually stopped and thought about it. We knew that sleep felt necessary. We knew that going without it made you miserable and eventually a little unhinged. But the actual machinery of it, the gears turning inside your brain right now as you drift closer to unconsciousness, that was a mystery for an astonishingly long time. And honestly, some of it still is. Let's start there. Let's start with what people used to think sleep was, because it tells you a lot about how strange and counterintuitive the real answer turned out to be. If you'd asked Aristotle, and people did ask Aristotle about basically everything, he would have told you that sleep was caused by warm vapors rising from your stomach after you ate a meal. The idea was that digestion produced these fumes, and the fumes traveled up to your brain, and your brain was cold, so the vapors condensed, and that heaviness made you fall asleep. It's a beautifully wrong theory. He laid it out around 350 BCE in a short work literally called On Sleep and Sleeplessness, which is probably the first dedicated scientific text about sleep in the Western tradition. And look, you can see where he was coming from. You eat a big meal, you feel drowsy. That's real. That's an observation anyone can make. He just got the mechanism completely backward. But give the man credit. He was at least asking the question. Most thinkers before him treated sleep as so obvious it didn't need explaining. You get tired, you sleep. What's to figure out? The thing is, that question, what is actually happening when you fall asleep, turns out to be one of the hardest questions in all of biology. And the reason it's so hard is that sleep doesn't look like it's doing anything. From the outside, a sleeping person is just lying there, not moving, not responding, not producing anything useful. And from an evolutionary standpoint, that's terrifying. Think about it. You're an animal on the savannah. Every hour you spend unconscious is an hour you're not finding food, not watching for predators, not mating, not doing any of the things that natural selection supposedly cares about. Sleep should have been eliminated millions of years ago. It's a vulnerability. It's a liability. And yet every animal with a nervous system does it, or something very close to it. Dolphins sleep with half their brain at a time so they can keep swimming. Migrating birds take microsleeps lasting a few seconds while they're flying. Fruit flies sleep. Even nematode worms, these tiny creatures with exactly 302 neurons, have sleep-like states. Whatever sleep is doing, it's so important that evolution has never found a way around it. Not once, in hundreds of millions of years. That should give you a sense of the stakes here. This isn't some minor biological curiosity. Sleep is doing something so fundamental, so non-negotiable, that every complex organism on the planet pays the enormous cost of unconsciousness rather than go without it. But for most of history, we couldn't even begin to investigate that question because we didn't have the tools. The brain was a black box. You could observe someone sleeping from the outside, and you could observe what happened when people didn't sleep, but you couldn't see what was happening inside. And that's where our story really begins. Not with the philosophers guessing about vapors, but with a German psychiatrist in the 1920s who figured out how to listen to the brain's electrical whisper. His name was Hans Berger. and he was, by most accounts, kind of an odd guy. Quiet, secretive, obsessively meticulous. He worked at the University of Jena, and for years, we're talking over a decade, he conducted experiments in near total secrecy because he was afraid people would think he was a crank. And here's the thing that motivated him, the thing that set him on this path. When Berger was a young man doing his military service, he had a horse riding accident. He fell off his horse and nearly got crushed by a cannon being pulled by a horse-drawn artillery unit. At that exact moment, the exact moment he was in danger, his sister, miles away, had a sudden, overwhelming feeling that something terrible had happened to him. She convinced their father to send a telegram asking if Hans was all right. This shook Berger deeply. He became convinced that the brain must generate some kind of energy, some kind of signal that could potentially be transmitted or at least detected. He wanted to find that energy. Now, he never did prove telepathy, obviously. But what he did find was arguably more important. In 1924, he successfully recorded the electrical activity of a human brain through the intact skull. He used silver foil electrodes, placed them on the scalp of a patient. His first subject was actually a young man who'd had a piece of skull removed during surgery, which made the signals easier to detect, and he connected those electrodes to a galvanometer, which is essentially a very sensitive device for measuring tiny electrical currents. And there it was, a rhythmic, oscillating signal, a wave. The brain was humming. He called it the electroencephalogram, the EEG, and he spent years quietly refining his technique, recording more and more subjects, before he finally published his findings in 1929. The scientific community's reaction was, let's call it, lukewarm. A lot of people didn't believe him. A psychiatrist claiming to read electrical signals from the brain through the skull? It sounded like exactly the kind of fringe science that serious researchers avoided. It took validation from Edgar Adrian, a Nobel Prize-winning physiologist at Cambridge, to convince the broader scientific world that Berger's recordings were real. Adrian repeated the experiments in 1934, confirmed the results, and suddenly the EEG was legitimate. And here's what matters for our story. Berger had noticed something in his recordings that would change everything we understand about sleep. When his subjects were awake and alert, the EEG showed fast, irregular, low-amplitude waves, lots of different brain regions firing at their own pace, doing their own thing. He called these beta waves. But when subjects closed their eyes and relaxed, something different appeared. The waves became slower, more regular, higher in amplitude. A smooth, rolling rhythm of about 8 to 12 cycles per second. He called these alpha waves. It was as if the brain, when it stopped actively processing the outside world, fell into a gentle, synchronized pulse, like an orchestra that had been playing a complex symphony, suddenly settling into a single, slow chord. That observation, that the brain's electrical activity changes in measurable, predictable ways depending on your state of consciousness, was the key that unlocked the science of sleep. Because if the brain does different things when you're awake versus when you're relaxed, what does it do when you're actually asleep? What happens to those waves when you cross the threshold into unconsciousness? It took a few more years for someone to really chase that question. In 1937, a scientist named Alfred Loomis, and Loomis is a fascinating character, a wealthy amateur scientist who built a private laboratory in his mansion in Tuxedo Park, New York. Loomis and his colleagues decided to do something nobody had done systematically before. They invited people to come to the lab and sleep while hooked up to EEG machines, and they recorded the brain's activity all night long. What they found was that sleep wasn't just one thing. It wasn't a uniform state of unconsciousness. The brain went through distinct stages, each with its own characteristic electrical signature. This was genuinely surprising. Before Loomis, the common assumption was that sleep was basically like turning off a light. You were either on or off, awake or asleep. But the EEG showed something far more interesting. As a person fell asleep, the brain waves got progressively slower and bigger. Loomis identified 5 stages, which he labeled A through E. Stage A was drowsiness, those alpha waves Berger had found, starting to break apart. Stage B was light sleep, where the alpha rhythm disappeared and was replaced by slower, more irregular activity. And then things got really interesting. In the deeper stages, C through E, the brain produced these massive, slow waves, big, rolling, oscillations that swept across the cortex like slow ocean swells. These were eventually called delta waves, and they cycle at just 1 to 4 times per second. Think about that. When you're awake, your brain is buzzing with activity at 20, 30, 40 cycles per second. In deep sleep, it slows down to 1 or 2. It's a profoundly different state. And here's what's really clever about what the brain is doing during those slow waves. But actually, let me come back to that in a moment, because there's another piece of the puzzle that was discovered about 15 years later, and it changes the whole picture. In 1953, a graduate student at the University of Chicago named Eugene Aserinsky was working in the lab of Nathaniel Kleitman. Now, Kleitman is sometimes called the father of modern sleep research, and for good reason. He'd been studying sleep since the 1920s, and he was one of the few scientists who took it seriously as a field of study when most of the medical establishment considered it a backwater. Kleitman was rigorous, demanding, and deeply committed to understanding the basic physiology of sleep. He'd even experimented on himself. In 1938, he and a colleague spent 32 days living in Mammoth Cave in Kentucky, 200 feet underground, in total darkness, trying to see whether the human sleep-wake cycle was driven by external cues like sunlight or by some internal clock. The answer, as it turned out, was the internal clock, but we'll get to that. So Aserinsky was Kleitman's graduate student, and he'd been given a somewhat unglamorous assignment, observing the eye movements of sleeping infants. And he noticed something peculiar. At certain points during sleep, the baby's eyes would start darting around rapidly under their closed eyelids. Quick, jerky movements, back and forth, as if they were watching something. This was odd. In most of sleep, the eyes are still or drift slowly. But these rapid movements were something different. Aserinsky mentioned this to Kleitman, and Kleitman, to his credit, didn't dismiss it. He told Aserinsky to look for the same thing in adults. They set up experiments. Adults sleeping in the lab, EEG electrodes on their scalps, and additional electrodes near their eyes to track eye movement. And sure enough, there it was. Periodically throughout the night, the sleeper's eyes would begin moving rapidly, and when Aserinsky looked at the EEG during these periods, he saw something that made no sense at first. The brain waves didn't look like deep sleep. They didn't even look like light sleep. They looked almost exactly like the brain waves of someone who was wide awake. Fast, low amplitude, desynchronized activity, the brain buzzing with energy. But the person was clearly asleep. Their muscles were relaxed. They didn't respond to sounds. They were, by every external measure, deeply unconscious. And yet their brain was lit up like Times Square. Aserinsky and Kleitman called this rapid eye movement sleep, REM sleep. And when they woke people up during these periods and asked what was happening, the answer was almost always the same: dreams, vivid, narrative, emotionally intense dreams. They'd stumbled onto the biological signature of dreaming. This discovery, published in 1953, completely rewrote the map of sleep. Because now it was clear that sleep wasn't a simple descent into unconsciousness and a climb back out. It was a cycle. You fall asleep. You go through progressively deeper stages of what we now call non-REM sleep. Those slow rolling delta waves getting bigger and slower, and then, after maybe 90 minutes, the brain does something extraordinary. It reverses course. The slow waves recede, the brain activity ramps back up to near waking levels, your eyes start darting around, and you enter this bizarre paradoxical state where your brain is wildly active, but your body is essentially paralyzed. You dream. And then, after maybe 10 or 20 minutes of REM, the whole cycle starts over. Deeper sleep, then back up to REM, then deeper again. You do this 4 or 5 times a night, and here's the detail that turns out to be really important. The composition of those cycles changes as the night goes on. Early in the night, the deep non-REM stages dominate. You spend long stretches in those slow delta waves. But as the night progresses, the deep sleep gets shorter and the REM periods get longer. By the early morning hours, you're spending most of your time in REM, dreaming elaborate dreams, your brain almost as active as it is during the day. And that pattern, that architecture, the way the cycles shift and evolve across a single night, it's not random. It's doing something specific. And, as it turns out, something that matters enormously for your memory, your emotional health, your immune system, and about a dozen other things you'd never guess were connected to how your brain behaves while you're lying there with your eyes closed, apparently doing nothing at all. But to understand what those different stages are actually accomplishing, we need to talk about what's happening at a much smaller scale. Not the sweeping electrical waves you can see on an EEG, but what individual neurons are doing, what chemicals are flooding certain parts of the brain and draining from others. Because the real story of sleep, the one that researchers have been piecing together for the last few decades, is a story about chemistry. And it starts with a molecule that's building up in your brain right now, this very moment. A molecule that's been accumulating since the instant you woke up this morning. And that molecule is called adenosine. And it's one of the most elegant little mechanisms in all of biology, because it works almost like an hourglass. A chemical hourglass that measures how long you've been awake. Here's how it works. Every second you're conscious, your brain cells are burning energy. Neurons are firing, processing information, keeping you alert, helping you think, helping you navigate the world. And the primary fuel they burn is a molecule called adenosine triphosphate, ATP. You might remember that name from a biology class somewhere. ATP is basically the universal energy currency of your cells. Every cell in your body uses it, but neurons are especially hungry. Your brain accounts for maybe 2% of your body weight, but it consumes roughly 20% of your energy. It's an expensive organ to run, and as neurons burn through ATP, one of the byproducts, one of the metabolic leftovers, is adenosine. Plain old adenosine, without the triphosphate part. And here's the key. Adenosine doesn't just float around doing nothing. It binds to specific receptors on neurons, and when it does, it has an inhibitory effect. It makes those neurons less likely to fire. It slows things down. It makes you feel drowsy. So think about what that means. The longer you've been awake, the more ATP your brain has burned, and the more adenosine has accumulated in the spaces between your neurons. Hour by hour, the adenosine builds up, and hour by hour, you get sleepier. It's a beautifully simple feedback loop. The act of being awake literally creates the pressure to sleep. Scientists call this sleep pressure, or more formally, the homeostatic sleep drive. And it's one of the two major systems that govern when you fall asleep and when you wake up. We'll get to the other one in a bit. But first, let's stay with adenosine for a moment, because there's a detail here that you're going to love. You already know a drug that works by interfering with this exact system. You probably had some this morning. Caffeine. That's what caffeine does. Caffeine molecules are shaped almost exactly like adenosine molecules, close enough that they fit into the same receptors on your neurons. But here's the trick. When caffeine binds to those receptors, it doesn't activate them. It just sits there, blocking the slot, like someone parking in a space without getting out of the car. The adenosine is still there, still accumulating, but it can't reach its receptors because caffeine is in the way. So you don't feel sleepy, you feel alert, focused, maybe a little wired. But the adenosine hasn't gone away. It's still building up in the background. And when the caffeine eventually gets metabolized, the half-life is roughly 5 to 6 hours, depending on your liver enzymes and genetics. When it clears out, all that accumulated adenosine suddenly floods into the now vacant receptors, and you crash. That afternoon wall of exhaustion that hits you if you had coffee at 7 in the morning. That's not new tiredness. That's all the tiredness you were supposed to feel gradually, hitting you at once. And this is why sleep researchers get a little uneasy about heavy caffeine use. It's not that caffeine is terrible for you. It's one of the most studied substances on the planet, and for most people, it's pretty benign. But it doesn't eliminate your sleep need. It masks it. You're borrowing alertness from your future self, and at some point, the bill comes due. The adenosine was there all along. Now, the person who really helped nail down how adenosine drives sleepiness was a researcher named Robert McCarley, working at Harvard Medical School in the 1970s and 80s, along with his colleague J. Allen Hobson. McCarley and Hobson did foundational work on the neurochemistry of sleep and waking states, and they proposed a model, the reciprocal interaction model, that described how different groups of neurons essentially take turns controlling your brain's state. And this is where the chemistry gets really interesting, because adenosine is just one player in a much bigger cast of characters. Here's the picture. When you're awake, certain regions of your brain are pumping out neurotransmitters that promote alertness. There's a cluster of neurons in the brainstem called the locus coeruleus. Beautiful name. It means the blue spot in Latin because the tissue actually looks bluish, and it releases norepinephrine, which keeps you alert and attentive. There's another group called the RAF nuclei that releases serotonin, which helps regulate mood and arousal. There's a region in the hypothalamus that releases histamine. And yes, that's the same histamine involved in allergies, which is exactly why antihistamines make you drowsy. They're dampening one of your brain's own wake-promoting signals. And then there's the basal forebrain, which releases acetylcholine, a neurotransmitter critical for attention and learning. So during the day, all these systems are active, firing away, keeping you conscious and engaged. But as adenosine builds up, it starts inhibiting these wake-promoting centers. It's like a dimmer switch slowly turning down the lights. The norepinephrine fades, the serotonin quiets, the histamine drops. And as those signals weaken, a different set of neurons gains the upper hand. Neurons in a region called the ventrolateral preoptic area, or VLPO, tucked in the front of the hypothalamus. The VLPO was identified as a critical sleep-promoting center by Cliff Saper and colleagues at Harvard in the mid-1990s. When the VLPO activates, it releases inhibitory neurotransmitters, GABA and galanin, that actively shut down all those wake-promoting centers. The locus coeruleus goes quiet. The Rafe nuclei go quiet. Histamine production drops to almost nothing, and you fall asleep. What's clever about this arrangement is that it works like a flip-flop switch, the kind of binary switch you'd find in an electronic circuit. You're either awake or asleep, with very little time spent in between. And that makes sense from a survival standpoint, right? You don't want to be half asleep. Half asleep is dangerous. You want a clean transition. Saper actually used that exact analogy. He called it a flip-flop circuit, and pointed out that the mutual inhibition between the wake centers and the VLPO creates a system that's stable in either state, but transitions rapidly between them. It's the same principle that makes a light switch snap on or off, rather than hovering in the middle. But here's where it gets even more interesting. There's a second system layered on top of all this adenosine-driven sleep pressure, and it operates on a completely different principle. It's not measuring how long you've been awake. It's measuring what time it is. And this is the circadian system, your internal clock. And the story of how we figured it out involves a cave, a bunker, and some very confused volunteers who had no idea what day it was. Remember Nathaniel Kleitman, the guy from the University of Chicago who mentored Eugene Aserinsky during the discovery of REM sleep? Well, back in 1938, 15 years before the REM discovery, Kleitman did something wonderfully eccentric. He wanted to know whether humans had a built-in clock, whether our bodies would maintain a regular sleep-wake cycle, even without any external cues like sunlight or clocks or social schedules. So he and a research assistant named Bruce Richardson went down into Mammoth Cave in Kentucky, the longest known cave system in the world, and they lived there for 32 days. The conditions were, by all accounts, miserable. It was damp. It was dark, obviously, and the temperature hovered around a constant 54 degrees Fahrenheit. They set up cots and tried to live on a 28-hour day instead of a 24-hour day to see if their bodies would adapt. Richardson, who was younger, adjusted somewhat. His body temperature and sleep patterns started to shift toward the 28-hour schedule. But Kleitman, who was in his early 40s, couldn't do it. His body stubbornly clung to something close to a 24-hour rhythm. His temperature would rise and fall on its own schedule, and he'd feel sleepy and alert at times that had nothing to do with the artificial schedule they'd set up. It was a suggestive result, but not definitive. The real breakthroughs in understanding the circadian clock came later, in the 1960s and 70s, when a German scientist named Jürgen Aschoff built an underground bunker, a time isolation facility, at the Max Planck Institute in Andechs, Bavaria. Aschoff and his colleague Rutger Wever recruited hundreds of volunteers over the years to live in this bunker for weeks at a time, completely cut off from natural light, clocks, television, radio, anything that could tell them what time it was outside. The volunteers could turn lights on and off, eat when they wanted, sleep when they wanted. Total freedom. And what Aschoff found was remarkable. Almost every single person settled into a regular, repeating cycle of sleep and wakefulness. But it wasn't exactly 24 hours. It was slightly longer, on average about 24 hours and 15 minutes, though it varied from person to person. Some people drifted to 25 hours, a few even longer. This meant that without external time cues, people would go to bed a little later each day and wake up a little later, slowly drifting out of sync with the actual day-night cycle outside. Aschoff called this a free-running rhythm, and it was powerful evidence that the body contains an internal oscillator, a genuine biological clock that runs on approximately, but not exactly, a 24-hour cycle. The word circadian comes from the Latin circa, meaning about, and dies, meaning day. About a day. That slight imprecision is actually important because it means the clock needs to be reset, entrained, in the technical language, every day by external cues, the most powerful of which is light. But where is this clock? What is it, physically? That question took decades to answer, and the answer turned out to be a tiny cluster of neurons, about 20,000 of them, sitting right above the point where your optic nerves cross on their way to the back of your brain. This crossing point is called the optic chiasm, and the cluster of neurons sitting just above it is called the suprachiasmatic nucleus, or SCN. And the SCN is, for all practical purposes, your master clock. It's the pacemaker that coordinates the timing of nearly every biological rhythm in your body, not just sleep, but hormone release, body temperature, digestion, immune function, even how sensitive your cells are to certain medications. The SCN was identified as the master circadian clock through a series of elegant experiments in the 1970s. In 1972, two independent research teams, one led by Robert Moore at the University of Chicago, the other by Irving Zucker at Berkeley, showed that destroying the SCN in rats abolished their circadian rhythms entirely. The animals didn't stop sleeping, but they slept in random fragments throughout the day and night, with no pattern at all. And then, in a really beautiful experiment in 1990, a researcher named Ralph transplanted SCN tissue from mutant hamsters that had an abnormally short circadian period of about 20 hours, into normal hamsters whose own SCN had been destroyed. And the recipient hamsters adopted the donor's rhythm. They started cycling on a 20-hour schedule. The clock wasn't just necessary for rhythms, it was sufficient. The timing information was right there in that little knot of cells. So now you've got two systems: the homeostatic drive, the adenosine hourglass, pushing you toward sleep the longer you've been awake, and the circadian clock, the SCN, telling your body what time of day it is and when sleep is appropriate. And these two systems interact in ways that are sometimes cooperative and sometimes surprisingly antagonistic. There's a model for this interaction that was developed by a sleep researcher named Alexander Borbély at the University of Zurich in 1982, and it's become one of the most influential frameworks in all of sleep science. Borbély called it the two-process model, and it's elegant in its simplicity. Process S is the sleep homeostat, the adenosine buildup. Process C is the circadian signal. And the interplay between them determines not just when you sleep, but how you feel throughout the day, and it explains some things that seem paradoxical at first. For instance, you'd think that sleepiness would just build and build throughout the day in a straight line, right? The longer you're awake, the sleepier you get. But that's not actually how it feels. Most people experience a dip in alertness in the early afternoon, that post-lunch slump around 1 or 2 o'clock. And then, strangely, they feel more alert again in the late afternoon and early evening, even though they've been awake even longer. How does that make sense? The adenosine is still accumulating. You should be getting sleepier. But the circadian signal is also rising in the late afternoon, actively promoting wakefulness, counteracting the adenosine. It's like the circadian system is giving you a second wind, propping you up just as the sleep pressure is getting heavy. And then, in the late evening, the circadian signal starts to drop. It begins allowing sleep. And suddenly, all that accumulated adenosine hits you like a wave, and you go from feeling reasonably alert to absolutely exhausted in what feels like minutes. This interplay also explains something that shift workers and new parents know all too well: the strange, almost hallucinatory quality of being awake at 3 or 4 in the morning. At that hour, both systems are working against you. The adenosine has been building for a long time, and the circadian clock is at its lowest ebb, actively promoting sleep. You're fighting both the hourglass and the clock. And that's why mistakes happen at that hour. Industrial accidents, medical errors, car crashes, the Chernobyl disaster, the Exxon Valdez oil spill, the 3 Mile Island nuclear incident, all occurred in the early morning hours, when the humans in charge were operating at the very bottom of their alertness curve. The engineering of sleep, you might say, has consequences far beyond the bedroom. But here's something that Borbély's model, as elegant as it is, doesn't fully explain. It tells you when you sleep and how much pressure you feel to sleep, but it doesn't really tell you what's happening during sleep that makes it so necessary. What is sleep actually doing that's so important? Your body will force you into it even when it's dangerous, even when there are predators around, even when you have a deadline. And to answer that question, researchers had to look much more closely at what's happening inside the brain during those different stages, the ones that Loomis and Oserinsky and Kleitman first mapped out with their EEGs, because it turns out that non-REM sleep and REM sleep are doing very different jobs, and the machinery involved is remarkably sophisticated, almost like 2 completely different workshops running in the same building on alternating shifts. Let's start with non-REM sleep, and specifically the deep kind, stages 3 and 4 in the old Loomis classification, what we now often just call slow-wave sleep. Remember those big rolling delta waves that Loomis first recorded in the 1930s? Those slow synchronized oscillations where huge populations of neurons are firing together, then going quiet, then firing together again, almost like a stadium doing the wave. It turns out those aren't just a signature of deep sleep. They're actually doing something. They're part of the mechanism. Here's what researchers started piecing together in the 1990s and 2000s. During slow-wave sleep, your neocortex, that's the outer layer of the brain, the part that handles your most sophisticated thinking, goes through these rhythmic cycles of activity and silence. The active phase is called the up state, when neurons are firing, and the quiet phase is called the down state, when they're essentially resting. And these alternate roughly once a second, which is why the EEG shows those slow 1 hertz-ish oscillations. Now here's what's clever about this. During those up states, the brain isn't just randomly firing. It's replaying things. Specifically, it's replaying patterns of activity that occurred during the day when you were awake and learning and experiencing things. This was first demonstrated convincingly in rats. In the 1990s, researchers at MIT, Matthew Wilson and Bruce McNaughton, were recording from neurons in the hippocampus of rats while the animals ran through mazes during the day. The hippocampus, as you might know, is a seahorse-shaped structure deep in the brain that's absolutely critical for forming new memories. When a rat runs through a maze, specific hippocampal neurons fire in specific sequences. This cell fires when the rat is at this corner. That cell fires when it reaches that junction, and so on. They're called place cells, and they essentially create a neural map of the experience. What Wilson and McNaughton discovered was that when those same rats fell asleep afterward, during slow-wave sleep, those exact same sequences of neural firing replayed. The same cells, in the same order, but compressed, running about 5 to 20 times faster than they had during the actual experience. It was as if the sleeping brain was playing back a sped-up recording of the day's events. And this wasn't random noise. The correlations were too precise, too consistent. The brain was literally rehearsing what it had learned. Now think about what that means. During the day, your hippocampus is rapidly encoding new experiences, new faces, new facts, new routes, new skills. It's like a scratch pad, jotting things down quickly. But the hippocampus has limited capacity, and the memories stored there are fragile. They're easily disrupted. For a memory to really stick, to become part of your long-term knowledge, it needs to be transferred to the neocortex, which has vastly more storage capacity and holds memories in a more stable, distributed form. And that transfer, that process of moving memories from the hippocampus to the neocortex and integrating them with everything you already know, that's what appears to be happening during slow-wave sleep. The technical term is memory consolidation, and the current model, developed substantially by researchers like Jan Born and his colleagues at the University of Tübingen in Germany, goes something like this. During the slow oscillations of deep sleep, the hippocampus replays its recently encoded memories during those upstates. At the same time, there are faster oscillations happening called sleep spindles. Those are bursts of activity at around 12 to 15 hertz that Loomis also noticed in his early recordings. And there are also sharp wave ripples in the hippocampus, very fast oscillations at around 80 to 100 hertz. All three of these rhythms, the slow oscillations, the spindles, and the ripples, become temporally coupled during deep sleep. They nest inside each other, like gears of different sizes meshing together. The ripples ride on the spindles, and the spindles ride on the up phase of the slow oscillation. And this coordinated rhythmic activity creates a channel, a pathway, through which information flows from the hippocampus into the neocortex. It's beautiful engineering, honestly. 3 different oscillatory systems, each generated by different brain structures, locking together with millisecond precision to shuttle information from temporary to permanent storage. And here's the thing. The degree to which these rhythms are well coordinated on any given night actually predicts how well you'll remember what you learned the day before. People who show tighter coupling between slow oscillations and sleep spindles perform better on memory tests the next morning. Born's group even showed that you can enhance this process artificially by applying very gentle electrical stimulation to the scalp during deep sleep, timed to boost the slow oscillations. They were able to improve people's memory for word pairs they'd learned before bed. You're not adding information. You're just turning up the volume on the brain's own consolidation machinery. So that's one major function of deep, non-REM sleep. Memory consolidation, the transfer and integration of new learning into long-term storage. But it's not the only thing happening during those quiet hours of slow-wave sleep. There's another discovery, much more recent, that genuinely changed how neuroscientists think about why we sleep. And it has to do with something almost embarrassingly physical: taking out the trash. In 2012, a researcher named Maiken Nedergaard at the University of Rochester made a discovery that, honestly, sounds almost too simple to be true. She and her team found that the brain has its own waste clearance system, a network of channels that flushes out metabolic byproducts, including toxic proteins. They called it the glymphatic system, a portmanteau of glial, because it relies on glial cells, specifically astrocytes, and lymphatic, because it functions somewhat like the lymphatic system in the rest of your body. Here's how it works. Cerebrospinal fluid, the clear liquid that surrounds your brain and spinal cord, gets pumped along channels that run alongside the brain's blood vessels. These channels are formed by the end feet of astrocytes, star-shaped glial cells that wrap around the blood vessels. The cerebrospinal fluid flows through the brain tissue, picks up waste products, metabolic debris, misfolded proteins, all the junk that accumulates from a day of intense neural activity, and then drains it out through the veins. It's essentially a plumbing system for your brain. But here's the part that made everyone sit up. Nedergaard's team discovered that this glymphatic system is dramatically more active during sleep than during wakefulness. Specifically, during slow-wave sleep, the interstitial spaces in the brain, the tiny gaps between cells, expand by about 60%. The cells literally shrink a little, opening up channels for fluid to flow through more freely. It's as if the brain is rearranging the furniture to let the cleaning crew in. And one of the key waste products that gets cleared during this process is beta amyloid, the protein that accumulates in the brains of Alzheimer's disease patients, forming those characteristic plaques. Think about that for a second. Every day, as your neurons fire and communicate and do the work of keeping you alive and thinking and feeling, they produce metabolic waste, including proteins that, if they build up, are toxic. And the primary time your brain cleans this waste out is during deep sleep. Which means that when you don't get enough deep sleep, when you're chronically sleep deprived, or when your sleep is fragmented, or when you're aging and your slow-wave sleep naturally diminishes, that waste accumulates. And there's now a growing body of evidence linking chronic sleep disruption to increased risk of Alzheimer's disease and other neurodegenerative conditions. It's not proven that poor sleep causes Alzheimer's. The relationship is almost certainly bidirectional, since Alzheimer's pathology itself disrupts sleep. But the correlation is strong enough to be genuinely concerning. So deep, non-REM sleep is doing at least two enormous jobs. It's consolidating your memories, transferring the day's learning from temporary hippocampal storage into the more permanent architecture of the neocortex, and it's flushing out the toxic metabolic waste that accumulated during the day. Both of these functions require those slow, synchronized oscillations, the delta waves, the same ones Loomis first saw on his ink traces in the 1930s. He had no idea what they were for. He just knew they were there. It took another 70 or 80 years before we started to understand that those big, lazy waves were the signature of the brain's most critical maintenance operations. Now that's deep, non-REM sleep. But what about the other kind? The strange, paradoxical state that Oserinsky stumbled onto when he noticed his son's eyes darting under closed lids. REM sleep, with its fast, wake-like brain activity, its vivid dreams, and its almost complete paralysis of the voluntary muscles. What's that doing? This is where things get, if anything, even more interesting, and honestly, a bit more mysterious. Because while the memory consolidation story for slow-wave sleep is fairly well established, the functions of REM sleep are still being actively debated. There are several strong theories, and they're not mutually exclusive. REM sleep is probably doing multiple things at once. But let me walk you through the main ones, because each of them reveals something fascinating about how your brain works when you're not paying attention to it. The first and perhaps most compelling theory connects REM sleep to emotional processing, and the person who's done more than almost anyone to develop this idea is Matthew Walker, the neuroscientist at UC Berkeley, whose popular book and research have brought a lot of sleep science into public awareness. Walker and his colleagues have shown that REM sleep appears to function as a kind of overnight therapy session. During REM, your brain reactivates emotional memories, experiences from the day that carried a strong emotional charge, but it does so in a neurochemical environment that's fundamentally different from waking life. And this is the key detail. During REM sleep, norepinephrine, that stress-related neurotransmitter we talked about, the one produced by the locus coeruleus, is almost completely shut off. The locus coeruleus goes quiet during REM. It's one of the only times in your entire life that your brain is processing emotional content in the absence of this particular stress chemical. Walker's theory is that this allows the brain to reprocess emotional memories and strip away some of their emotional intensity while preserving the informational content. You remember what happened, but the sting fades. The visceral, gut punch quality of a painful experience gets softened. And there's experimental evidence for this. People who get adequate REM sleep show reduced amygdala reactivity to previously upsetting images the next day, compared to people who were deprived of REM specifically. The memory is intact, but the emotional edge has been filed down. It's as if REM sleep is saying, yes, remember this happened. Learn from it. But you don't need to keep feeling it at full intensity forever. And this connects to something clinicians have observed for a long time about post-traumatic stress disorder. People with PTSD often have disrupted REM sleep. They wake frequently from REM, or their REM periods are fragmented, or, intriguingly, their norepinephrine levels remain elevated even during REM, which shouldn't happen. The stress chemical that's supposed to go quiet doesn't go quiet, and so the emotional memories keep getting replayed at full intensity, night after night, never getting that therapeutic reprocessing. The nightmares don't fade. The emotional charge never diminishes. There's actually a blood pressure medication called prazosin that blocks norepinephrine receptors, and it's been used with some success to reduce nightmares in PTSD patients, essentially doing pharmacologically what healthy REM sleep should be doing naturally. But emotional processing isn't the only thing REM sleep seems to be up to. There's another function that's arguably just as important, and it has to do with creativity, with the way your brain finds hidden connections between ideas that seem totally unrelated on the surface. Think about what your brain is doing during REM. It's highly active. Metabolically, it looks almost like you're awake. But the prefrontal cortex, the part responsible for logical, linear, rule-following thought, is dialed way down. Meanwhile, the associative areas of your cortex, the regions that make loose lateral connections between disparate concepts, are firing like crazy. It's as if your brain has taken the librarian who carefully files everything in the right category and sent her home for the night, and instead turned the whole library over to some brilliant, slightly unhinged artist who's pulling books off random shelves and stacking them next to each other just to see what happens. And sometimes what happens is genuinely extraordinary. There's a famous study from 2004 by a group at the University of Lübeck in Germany, led by Ulrich Wagner and Jan Born. The same Born, whose group we talked about earlier with the slow oscillation work. They gave subjects a math task that had a hidden shortcut. The task was solvable the long way, step by step, but there was an elegant trick buried in the structure that, if you noticed it, would let you skip most of the work. They tested people after 8 hours of being awake, and about 20% of them spotted the shortcut. Not bad. But people who got a full night's sleep before being retested, nearly 60% found it. 3 times as many. Sleep didn't just consolidate what they already knew. It reorganized the information in a way that revealed a pattern they hadn't consciously detected. And here's what's really interesting. It wasn't just any sleep that did this. The effect was specifically correlated with the amount of REM sleep the subjects got. More REM, more insight. And this makes intuitive sense when you think about what REM is doing neurologically. With the prefrontal cortex quieted and the associative cortex running wild, your brain is essentially free associating through everything it's absorbed recently, testing unlikely combinations, linking a memory from Tuesday with a concept from 6 months ago, with a sensation from childhood. Most of these combinations are nonsense, which is why dreams are so often bizarre and incoherent. But every once in a while, one of those random juxtapositions lands on something real, something your waking brain would never have tried because it would have seemed too unlikely, too far-fetched. This is probably why so many creative breakthroughs have famously come during or just after sleep. You've heard some of these stories. Kekulé claiming the ring structure of benzene came to him in a dream of a snake biting its own tail. Mendeleev reportedly dreaming the arrangement of his periodic table. Paul McCartney saying the melody for Yesterday arrived complete in a dream. Now, some of these stories are likely embellished. People love a good eureka narrative, and memory is unreliable. But the underlying principle is solid. REM sleep creates a neurochemical environment uniquely suited to finding hidden connections that logical, waking thought would filter out as improbable. So let's step back for a moment and appreciate what we've covered about what sleep actually does. During deep, non-REM sleep, your brain is consolidating memories, replaying the day's experiences, transferring important information from temporary storage in the hippocampus to more permanent homes in the cortex. It's also physically cleaning itself. The glymphatic system is flushing out metabolic waste, including the beta-amyloid proteins associated with Alzheimer's. Then, during REM sleep, your brain is doing emotional maintenance, reprocessing difficult experiences in the absence of norepinephrine so their emotional charge can diminish. And it's doing creative recombination, testing wild associations between stored memories and concepts. These are profoundly different functions, happening in different stages, using different neurochemistry, and they're all essential. Skip any one of them, and something goes wrong. Which brings us to something that affects an enormous number of people, and that sleep science has spent decades trying to understand. What happens when sleep goes wrong? Not just one bad night, but chronically. What happens to a human body and a human brain when sleep is consistently insufficient or consistently disrupted? And the answer, frankly, is that almost everything goes wrong. The breadth of it is staggering. Let's start with the cognitive effects, because they're the most immediately obvious and the most well-studied. In 2003, a research group at the University of Pennsylvania, led by Hans van Dongen and David Dingus, published what became one of the most cited sleep deprivation studies in history. They took healthy adults and divided them into groups. Some slept 8 hours a night. Some slept 6. Some slept 4. And one unlucky group stayed awake for 3 consecutive days. This went on for 2 weeks. Well, except for the total deprivation group, obviously, who were monitored for their 3 days straight. Every day, everyone took a battery of cognitive tests measuring things like reaction time, attention, working memory. Here's what they found. The people sleeping 8 hours stayed sharp throughout the entire 2 weeks. No decline. The people sleeping 4 hours deteriorated rapidly and dramatically. Within a few days, Their cognitive performance was as bad as someone who'd been awake for 2 or 3 days straight. But here's the really unsettling part. The finding that changed how researchers think about sleep debt. The people sleeping 6 hours, which, let's be honest, describes a huge portion of the adult population. They declined too. More slowly, more subtly, but steadily, night after night, with no plateau. After 2 weeks of 6-hour nights, their cognitive performance was statistically equivalent to someone who had been totally sleep-deprived for 24 hours straight. They were as impaired as someone who'd pulled an all-nighter. But, and this is the kicker, they didn't feel impaired. When asked to rate their own sleepiness and their own performance, they consistently said they were doing fine. They had habituated to their diminished state. They'd lost the ability to accurately perceive how impaired they actually were. Think about what that means. Millions of people walking around right now, driving cars, making medical decisions, operating machinery, managing other people's money, flying airplanes, functioning at the cognitive level of someone who hasn't slept in 24 hours, and genuinely believing they're fine. This is one of the most dangerous findings in all of sleep science. It's not just that insufficient sleep makes you worse at things. It's that it simultaneously makes you unable to tell that you're worse at things. You lose the very faculty you'd need to recognize the problem. And the effects aren't limited to attention and reaction time. Working memory suffers. Decision-making degrades, particularly decisions involving risk assessment. Sleep-deprived people tend to make riskier choices and weigh potential rewards more heavily than potential losses. Emotional regulation deteriorates. The amygdala becomes hyper-reactive, while its connection to the prefrontal cortex weakens, which is essentially the neural recipe for overreacting to everything. Matthew Walker's lab at Berkeley did brain imaging studies showing that sleep-deprived subjects had roughly 60% greater amygdala reactivity to negative emotional stimuli compared to well-rested subjects. 60%. That's not a subtle shift. That's a fundamentally different emotional landscape. And then there are the physical consequences, which accumulate more slowly, but are arguably even more alarming. Chronic sleep restriction, even modest restriction, like averaging 6 hours instead of 8, is associated with increased risk of cardiovascular disease, type 2 diabetes, obesity, and impaired immune function. The mechanisms are varied and interconnected. Sleep loss elevates cortisol, the stress hormone, particularly in the evening when it should be declining. It reduces insulin sensitivity, meaning your cells become less responsive to insulin and your blood sugar regulation starts to falter. It increases levels of ghrelin, the hunger hormone, while decreasing leptin, the satiety hormone, so you feel hungrier and less satisfied after eating, which is a pretty direct pathway to weight gain. It suppresses the activity of natural killer cells, which are a critical part of your immune system's ability to identify and destroy abnormal cells, including early-stage cancer cells. There was a striking study, I think it was Sheldon Cohen's group at Carnegie Mellon, published around 2009, where they actually exposed healthy volunteers to the common cold virus via nasal drops and then tracked who got sick. The single strongest predictor of who developed a clinical cold wasn't age, wasn't stress level, wasn't body mass index, wasn't baseline antibody levels. It was how much they'd been sleeping in the preceding 2 weeks. People averaging less than 7 hours of sleep were nearly 3 times more likely to develop a cold than people sleeping 8 hours or more. 3 times. From 1 hour's difference. And the cardiovascular effects are just as sobering. There's a natural experiment that happens twice a year across most of North America and Europe that researchers have used to study this. Daylight saving time. In the spring, when clocks move forward and everyone loses an hour of sleep, hospitals consistently see a measurable spike in heart attacks the following Monday, somewhere around 20 to 25%, depending on the study. In the fall, when clocks move back and people gain an hour, heart attacks drop by a similar margin. 1 hour, one single hour of sleep across a population, is enough to move the needle on cardiac events. That's how thin the margins are. Now, all of this research, the cognitive studies, the immunological work, the cardiovascular epidemiology, it all converges on a picture that would have been unimaginable to scientists even 50 years ago. Sleep isn't rest. Sleep isn't the absence of wakefulness. Sleep is an active, complex, multi-stage biological process that maintains virtually every system in your body, and when it's compromised, the consequences ripple outward into every domain of health and function. The old macho attitude of, "I'll sleep when I'm dead," turns out to be, in a very literal sense, self-fulfilling. But understanding that sleep matters and understanding how to fix it when it breaks are two very different things. And for most of the 20th century, medicine's primary answer to disordered sleep was pharmacology, sleeping pills. The history of how we tried to drug ourselves to sleep is its own fascinating and often troubling story, and it starts in a place you might not expect. It starts with barbiturates. Specifically, it starts in 1864 in the laboratory of a German chemist named Adolf von Baeyer. Now, Baeyer would go on to win the Nobel Prize in Chemistry in 1905 for his work on organic dyes, but decades before that, as a young researcher, he synthesized a compound by combining urea, a waste product found in urine, with malonic acid, which comes from apples. The resulting substance was barbituric acid. And here's a small, slightly amusing historical footnote. Nobody is entirely sure where the name barbituric came from. One popular story says Baeyer named it after a woman named Barbara, possibly a girlfriend or a patron saint celebrated on the day of the discovery. Another version claims he celebrated the synthesis at a tavern frequented by artillery officers, and Barbara was the patron saint of artillerymen. The truth is probably lost to time. But the compound itself, barbituric acid, wasn't particularly useful on its own. It had no real sedative properties. It just sat there in the chemical literature for about 4 decades, waiting. Then, in 1903, 2 German chemists named Emil Fischer and Joseph von Mering figured out how to modify barbituric acid into something pharmacologically powerful. They synthesized diethylbarbituric acid, and they marketed it under the trade name Veronal. And Veronal worked. It really worked. It put people to sleep. Fischer and von Mering had essentially created the first commercially successful sleeping pill, and it was a sensation. Doctors prescribed it widely, patients loved it, and for a while, it seemed like modern chemistry had solved the problem of insomnia. But here's the thing about barbiturates, and this is a pattern you'll see repeated throughout the history of sleep pharmacology. They didn't actually produce normal sleep. What they did was suppress neural activity broadly and powerfully. They enhanced the effect of GABA, the brain's main inhibitory neurotransmitter, essentially turning down the volume on huge swaths of brain function. The result looked like sleep from the outside. The person was unconscious. They were still. They weren't responding to stimuli. But the internal architecture of that unconsciousness was profoundly different from natural sleep. Barbiturates suppressed REM sleep significantly. They altered the proportions of the different non-REM stages. They produced something closer to a chemical sedation than to the carefully orchestrated neurological process we've been talking about. The one with the slow oscillations, coupling to spindles, coupling to ripples. The one with the glymphatic channels opening up. The one with the emotional reprocessing happening in REM. And the problems didn't stop there. Barbiturates were addictive. Profoundly, dangerously addictive. Your body developed tolerance quickly, meaning you needed higher and higher doses to achieve the same effect. And the withdrawal could be genuinely life-threatening. Seizures, delirium, even death. Worse still, the therapeutic window was narrow. The dose that put you to sleep wasn't that far from the dose that could kill you. Barbiturate overdose became one of the leading methods of suicide in the mid-20th century. Marilyn Monroe's death in 1962, ruled a probable suicide, involved an overdose of Nembutal, which was a barbiturate. Judy Garland's death in 1969 was linked to barbiturate use. These weren't obscure chemicals. They were sitting in medicine cabinets across the Western world, prescribed by well-meaning doctors who didn't yet have better options. So the search was on for something safer. And in the 1960s, along came a new class of drugs, the benzodiazepines. The first one to really break through was chlordiazepoxide, marketed as Librium in 1960, followed by diazepam, you know it as Valium, in 1963. These were developed by Leo Sternbach, a chemist working at Hoffman La Roche in New Jersey. Sternbach had actually synthesized the core compound years earlier and set it aside as unpromising. It was only rediscovered by accident when a colleague was cleaning out the lab and found an untested sample. They ran it through pharmacological screening almost as an afterthought, and it turned out to be remarkably effective as an anxiolytic and sedative. Valium became one of the most prescribed drugs in the world by the 1970s. Between 1968 and 1982, it was the single best-selling pharmaceutical in the United States. Doctors prescribed it for anxiety, for muscle tension, for insomnia, for practically anything that seemed stress-related. And benzodiazepines were genuinely safer than barbiturates. The therapeutic window was much wider, meaning it was much harder to fatally overdose on them alone. But, and you probably sense a but coming, they shared many of the same fundamental problems. They also worked by enhancing GABA activity, though at a different binding site on the GABA-A receptor. They also suppressed certain stages of sleep, particularly reducing slow-wave deep sleep and altering sleep architecture. They also caused dependence, though the withdrawal was typically less lethal than barbiturate withdrawal. And they also didn't produce anything close to natural sleep. The person taking Valium or one of its cousins might feel like they slept, and they might not remember waking up during the night. Benzodiazepines can cause anterograde amnesia, meaning you don't form memories of the periods when you're under their influence. But the restorative processes that depend on proper sleep architecture were still being disrupted. Now you might be thinking, okay, but surely modern sleeping pills are better. And the answer is, somewhat. The next generation arrived in the 1990s with the so-called Z drugs, Zolpidem, which you know as Ambien, Zaloplon, marketed as Sonata, and Zopiclone, sold as Imovane or Lunesta in its refined form as Zopiclone. These drugs were designed to be more selective in how they targeted the GABA-A receptor, binding preferentially to a specific subunit called alpha-1, which is more directly associated with sedation. The idea was that by being more targeted, they'd produce fewer side effects and something closer to natural sleep. And in some ways, they did represent an improvement. They had shorter half-lives, meaning they cleared your system faster, which reduced next-day grogginess. They were somewhat less prone to causing dependence, though dependence was still absolutely possible. But here's what's really interesting, and a little sobering. When researchers like Matthew Walker and Daniel Kripke examined the actual sleep architecture produced by Z-drugs, the picture wasn't as rosy as the marketing suggested. Zolpidem still reduced sleep spindle activity. It still altered the natural proportions of sleep stages. A study Kripke published in 2012, drawing on data from over 30,000 patients, found that people prescribed sleeping pills, even modern ones, had significantly higher mortality rates than matched controls who weren't taking them. Now, that's an observational finding, and it doesn't prove the pills caused the deaths. People who are prescribed sleeping pills are often sicker in other ways, more stressed, dealing with more health problems. The causation question is genuinely complicated, but the association was striking enough that it raised serious questions about whether we should be prescribing these drugs as freely as we do. And this is where the story takes a turn that I find genuinely hopeful. Because while pharmacology was stumbling through this cycle of promise and disappointment, barbiturates to benzodiazepines to Z-drugs, each generation a little better, but none truly solving the problem, a completely different approach to insomnia was quietly being developed. It didn't involve any drugs at all. It came out of cognitive behavioral psychology, and it's called CBTI, Cognitive Behavioral Therapy for Insomnia. The roots of CBT go back to the 1970s and 1980s, when researchers like Arthur Spielman at City College of New York and Charles Morin at Université Laval in Quebec started thinking about insomnia not as a chemical problem requiring a chemical solution, but as a behavioral and cognitive problem, a set of learned habits and thought patterns that perpetuate wakefulness. Think about what happens when someone develops chronic insomnia. They have a few bad nights, maybe triggered by stress or jet lag or illness. Perfectly normal. But then they start worrying about sleep. They start going to bed earlier, trying to catch up. They lie in bed for hours, staring at the ceiling, and their brain begins to associate the bed with wakefulness and anxiety rather than with sleep. They start checking the clock, calculating how many hours they have left, getting more anxious with each passing minute. They might start napping during the day, which reduces their sleep pressure. Remember the adenosine system we talked about, the homeostatic drive, and makes it even harder to fall asleep at night. A vicious cycle forms. The insomnia becomes self-sustaining, independent of whatever originally triggered it. What Spielman and Morin and their colleagues realized was that you could break this cycle not with drugs, but with structured behavioral interventions. The core techniques are deceptively simple. Sleep restriction therapy, for instance, sounds almost cruel. You actually limit the amount of time the person spends in bed to match only the amount of time they're actually sleeping. If someone is lying in bed for 9 hours but only sleeping 5, you restrict their time in bed to 5 hours. This builds up enormous sleep pressure, all that adenosine accumulating, and it also retrains the brain to associate the bed with sleep rather than with lying awake. Then you gradually extend the window as sleep efficiency improves. Stimulus control is another key technique. You use the bed only for sleep. You get out of bed if you're awake for more than about 15 or 20 minutes. You go to another room and do something quiet until you feel sleepy again. The goal is to rebuild the association between bed and sleep. And then there's the cognitive component, addressing the catastrophic thinking that so often accompanies insomnia. Thoughts like, if I don't fall asleep in the next 30 minutes, tomorrow will be ruined. Or, I haven't slept properly in weeks. My health must be deteriorating. These thoughts are understandable, but they're also counterproductive, because anxiety is one of the most potent wake-promoting signals your brain can generate. It activates the locus coeruleus. It floods the system with norepinephrine. It's the exact opposite of what you need to trigger that VLPO flip-flop switch into sleep mode. CBTI teaches people to recognize these thought patterns and reframe them, not by pretending everything is fine, but by developing a more realistic and less catastrophic relationship with the occasional bad night. Here's what's remarkable. When you look at the clinical evidence, and there are now dozens of randomized controlled trials and several meta-analyses, CBTI is at least as effective as sleeping pills for short-term treatment of insomnia, and it's significantly more effective in the long term. A landmark study published in 2004 in the Journal of the American Medical Association compared CBT1 to zolpidem over an 8-week treatment period and then followed patients after treatment ended. During the active treatment phase, both approaches worked comparably well, but after treatment stopped, the CBTI group maintained their improvements while the drug group relapsed. The sleep gains from behavioral therapy were durable in a way that pharmacological sleep simply wasn't. In 2016, the American College of Physicians issued a clinical practice guideline recommending CBT1 as the first-line treatment for chronic insomnia in adults, ahead of any medication. Not as an alternative, as the preferred approach. And yet, despite this recommendation, the vast majority of people with insomnia still receive pills rather than therapy. The reasons are mostly practical. CBTI requires trained therapists, multiple sessions over several weeks, and active participation from the patient. A prescription can be written in 60 seconds. There aren't nearly enough trained CBTI providers to meet demand. This gap has driven a lot of interest in digital CBTI, app-based and online programs that deliver the same techniques through guided modules. Some of these, like the program developed by Colin Espy at the University of Oxford, have shown promising results in clinical trials. But we're still in the early stages of figuring out how to scale this kind of intervention. Now, the pharmacology story isn't over, because there's a newer class of sleep drugs that works on an entirely different principle. And it's worth understanding, because it reflects how much our knowledge of sleep neuroscience has advanced. These are the orexin receptor antagonists, drugs like suvorexant, marketed as Belsomra, approved by the FDA in 2014, and lemborexant, marketed as Divigo, approved in 2019. Remember the orexin system? Those neurons in the lateral hypothalamus that we touched on earlier, the ones that promote wakefulness and stabilize the flip-flop switch, the ones whose loss causes narcolepsy. These new drugs work by blocking orexin receptors, essentially quieting the wake promotion signal rather than artificially amplifying the sleep signal. It's a fundamentally different philosophy. Instead of sedating the brain into unconsciousness, you're removing one of the barriers to the brain's own natural sleep initiation process. Early evidence suggests these drugs may preserve sleep architecture more faithfully than their predecessors, though the long-term data is still accumulating. But perhaps the most exciting frontier in sleep medicine isn't about treating insomnia at all. It's about something much more fundamental, understanding the individual differences in how people sleep and why. Because one of the things that's become increasingly clear over the past 2 decades is that there is no single correct way to sleep. The 8-hour recommendation you hear everywhere, it's a population average, and the variation around that average is enormous. Some of this variation is genetic, and the story of how we started to unravel the genetics of sleep timing begins with a remarkable family that walked into a clinic in the late 1990s, complaining about something that might not sound like a problem at all. They kept waking up incredibly early. We're not talking about being a morning person. We're talking about people who were wide awake and alert at 3:30 in the morning, who were eating dinner at 4:30 in the afternoon, who were falling asleep by 7 in the evening no matter what they tried. Their entire daily rhythm was shifted forward by about 4 hours compared to the rest of the world. And this pattern ran in the family. Grandmother, mother, daughter, multiple relatives across generations, all locked into this dramatically advanced sleep schedule. The clinician who took this seriously was a neurologist named Louis Ptacek, working at the University of California, San Francisco. Ptacek had a background in genetics, and when he heard about this family, he recognized something potentially profound. This wasn't just a lifestyle choice or a habit. This had the hallmarks of a heritable trait, a Mendelian pattern, passed down through generations in a way that suggested a single gene might be responsible. So Ptacek and his colleague Ying-Hui Fu began the painstaking work of genetic analysis, collecting blood samples from affected and unaffected family members, mapping their genomes, looking for the mutation that set these extreme early birds apart. And in 2001, they found it. A single-point mutation in a gene called PER2, one of the core clock genes, the very molecular gears of the circadian system we talked about earlier. Remember the transcription-translation feedback loop running in every cell? The one that takes roughly 24 hours to complete one cycle. PER2 is a critical component of that loop. The mutation they found changed just one amino acid in the PER2 protein, at a site where the protein normally gets tagged with a phosphate group by an enzyme called casein kinase 1. That phosphate tag is part of what controls how quickly the PER2 protein accumulates, enters the nucleus, and shuts down its own gene's activity. Without that tag being placed correctly, the whole cycle runs faster. The clock still ticks, but each tick takes slightly less time. The 24-hour cycle becomes more like a 20-hour cycle, and the body's internal day ends earlier, which means sleep onset and wake time both shift dramatically forward. Think about what that means. A single letter change in the genetic code, one amino acid swapped for another, and your entire experience of time, when you feel alert, when you feel hungry, when you feel sleepy, shifts by hours. It's a beautiful and slightly unsettling demonstration of how precisely these molecular clocks govern our lives. This condition was named Familial Advanced Sleep Phase Syndrome, or FASPS, and it opened the floodgates. Because once you know that clock genes can be mutated in ways that shift sleep timing, you start looking for other variants, and you start finding them. Fu and Ptacek went on to discover additional mutations in other clock genes, CK1 delta, another component of that phosphorylation machinery, and others, each producing its own distinctive shift in sleep timing. And on the other end of the spectrum, researchers began identifying genetic variants associated with extreme night owls, people whose clocks run longer than 24 hours and who naturally drift toward later and later bedtimes. Some of these variants affect the same clock genes, but in different ways, lengthening the cycle rather than shortening it. Here's what's really important about all of this, and it's something that doesn't get enough attention. For decades, society has treated sleep timing as a moral issue. Early risers are disciplined, productive, virtuous. Night owls are lazy, undisciplined, probably up to no good. Benjamin Franklin's early to bed, early to rise gets quoted as if it's a law of nature rather than the preference of one particular 18th century polymath. But the genetics tells us something completely different. Your chronotype, whether you're naturally a morning person or an evening person, is substantially biological. Studies of twins suggest that somewhere between 30 and 50% of the variation in chronotype is heritable. You didn't choose to be a night owl any more than you chose your eye color. And forcing a strong night owl into an early morning schedule isn't character building. It's a form of chronic circadian misalignment that has real health consequences. This connects to something called social jet lag, a term coined by the German chronobiologist Till Roenneberg around 2006. Social jet lag is the discrepancy between your biological clock and the schedule society imposes on you. If your body naturally wants to fall asleep at 1 in the morning and wake at 9, but your job requires you to be at a desk by 8, you're setting your alarm for 6:30. On workdays, you're essentially living in the wrong time zone. You're perpetually jet-lagged, not because you flew anywhere, but because the social clock and your biological clock are mismatched. On weekends, you revert to your natural rhythm, sleeping late to pay back some of that debt, and then Monday morning hits like a transatlantic flight. Roenneberg's research, based on massive surveys of sleep timing across populations, showed that social jet lag affects a huge proportion of the population and correlates with higher rates of obesity, depression, cardiovascular problems, and poor academic performance. It's not a trivial inconvenience. It's a public health issue hiding in plain sight. And this brings us to one of the most practical and contentious applications of sleep science: school start times. Because if there's one population that suffers disproportionately from social jet lag, it's adolescents. And the biology here is really clear. During puberty, the circadian clock undergoes a well-documented shift. The onset of melatonin secretion, the signal that tells the brain it's time to start winding down, moves later by 1 to 3 hours compared to pre-pubertal children. This isn't teenagers being rebellious or irresponsible. It's a biological phase delay. Their brains are literally not ready for sleep until later at night, which means they're not ready to wake up until later in the morning. And yet, across much of the United States and many other countries, high schools start earlier than elementary schools, sometimes as early as 7 or 7:15 in the morning, which means teenagers are being asked to perform cognitively demanding tasks at a time when their biology says they should still be asleep. The researcher who did more than almost anyone to bring this issue to public attention is Mary Carskadon at Brown University. Starting in the 1990s, Carskadon conducted a series of studies showing that adolescents have a genuine biological need for about 8.5 to 9.5 hours of sleep, that their circadian phase is delayed relative to younger children, and that early school start times result in chronic sleep deprivation for the majority of teenagers. Her work showed that students in schools starting before 8 in the morning were significantly more likely to be sleep deprived, to report symptoms of depression, to have higher rates of car accidents, and to perform worse academically than students in schools with later start times. Now, you might think, this would be an easy policy fix. The science is clear. The intervention is straightforward. Just start school later. But the reality, as with so many things, is complicated by logistics, economics, and inertia. Bus schedules, after-school sports, parents' work schedules, childcare arrangements. All of these create resistance to change. And yet, the evidence kept accumulating. In 2014, the American Academy of Pediatrics issued a policy statement recommending that middle and high schools start no earlier than 8:30 in the morning, calling insufficient sleep in adolescents an important public health issue. California became the first state to legislate later school start times in 2019, requiring high schools to begin no earlier than 8:30. and early data from districts that have made the switch has been encouraging. More sleep, better attendance, fewer tardies, improved mood, and in some cases, measurable improvements in academic performance. It's a beautiful example of how basic science, understanding the molecular biology of circadian clocks, understanding the physiology of adolescent sleep, can eventually translate into policy that affects millions of lives. But it took decades. Carskadon was publishing this work in the 90s, and we're still fighting about it in many places. So let's zoom out for a moment and think about where all of this leaves us. We've traced the science of sleep from Aristotle's speculation about digestive vapors all the way to molecular genetics and public health policy. We've watched the field move from complete ignorance about what happens during sleep to a detailed understanding of sleep stages, neurotransmitter systems, circadian clocks, memory consolidation, waste clearance, emotional processing, and genetic variation. And yet, and here's the thing that keeps sleep researchers up at night, if you'll forgive the irony, the most fundamental question of all remains stubbornly unanswered. We still don't have a complete unified theory of why we sleep. We have pieces, excellent pieces. Memory consolidation is clearly part of the answer. Metabolic restoration is part of it. Glymphatic clearance is part of it. Emotional regulation is part of it. Immune function, synaptic homeostasis, energy conservation, all part of it. But no single theory accounts for everything. And the reason this matters isn't just academic. It matters because without a unified understanding, we're still making educated guesses about some very practical questions. How much sleep does a given individual actually need? What exactly are the consequences of losing specific types of sleep? Can you ever truly recover from chronic sleep deprivation, or is some of the damage permanent? That last question has gotten increasingly urgent attention in recent years, and some of the emerging answers are sobering. There's a growing body of research suggesting that chronic sleep loss may have cumulative effects on the brain that aren't fully reversible. Remember the glymphatic system, the brain's waste clearance mechanism that's most active during deep sleep? If you're consistently not getting enough deep sleep, beta amyloid and tau proteins accumulate. And once those proteins reach certain concentrations, they can begin triggering inflammatory cascades and neuronal damage that may not be undone simply by catching up on sleep later. This is still an active area of research, and we should be careful not to overstate what we know, but the direction of the evidence is concerning enough that it's changed how many sleep scientists talk about chronic sleep restriction. It's not just about feeling tired the next day. It may be about long-term brain health in ways we're only beginning to appreciate. And this connects to one of the most active frontiers in sleep research right now, the relationship between sleep and neurodegenerative disease. We touched on the Alzheimer's connection earlier, but the story extends beyond Alzheimer's. Disrupted sleep is a feature of Parkinson's disease, Lewy body dementia, and several other neurodegenerative conditions, and in many cases, the sleep disruption appears years or even decades before the other symptoms. Which raises a tantalizing and terrifying question. Is poor sleep a cause of neurodegeneration, a consequence of it, or both? The answer, increasingly, seems to be both, a vicious cycle where early disease pathology disrupts sleep, and disrupted sleep accelerates the disease pathology. If that's true, then sleep might be one of the earliest intervention points for diseases we currently can't treat effectively once they're established. This is why you're seeing an explosion of interest in sleep monitoring, sleep optimization, and sleep biomarkers in the medical research community. The idea that a wearable device on your wrist might one day detect subtle changes in your sleep architecture that predict Alzheimer's risk decades before cognitive symptoms appear. That's not science fiction anymore. It's an active research program at multiple major universities. Whether it pans out remains to be seen, but the underlying logic is sound, and the stakes are enormous. But there's another dimension to individual sleep differences that we haven't touched on yet, and it's one that challenges some of our most basic assumptions about what normal sleep even looks like. Because everything we've discussed so far has assumed a particular model of sleep. The consolidated monophasic block of 7 to 9 hours that modern Western society treats as the default. One long stretch of sleep at night, one long stretch of wakefulness during the day. And for most of us, that feels so natural, so obviously correct, that we never question it. But the historical and anthropological evidence suggests that this pattern might be a relatively recent invention, something our bodies were never really designed for. The person who brought this idea into the mainstream was a historian named Roger E. Kirch at Virginia Tech. In 2001, he published a paper, and then in 2005, a full book called At Day's Close, Night in Times Past, and what he found was remarkable. Kirch had spent 16 years combing through historical documents, diaries, court records, medical texts, prayer books, literature, letters, more than 500 references spanning centuries and multiple cultures. And what kept showing up again and again was a pattern of sleep that looked nothing like our consolidated 8-hour block. People referred to first sleep and second sleep as casually as we might say breakfast and lunch. It was just how things were. You'd go to bed not long after dark, sleep for 3 or 4 hours, then wake up for a period, sometimes an hour, sometimes 2, and then fall back asleep for another 3 or 4 hours, until dawn. That wakeful period in the middle? It had a name. In French it was called la dorveille, the wake sleep. And people used it for all sorts of things. They'd pray, they'd reflect on dreams, they'd talk quietly with a spouse, they'd get up and do small chores, they'd visit neighbors. Some medical texts from the 16th century actually recommended that couples trying to conceive should use this interval because the body would be rested and relaxed. It wasn't treated as insomnia. It wasn't treated as a problem at all. It was just the normal shape of a night. And here's what's really fascinating. Kirch noticed that references to first and second sleep began to disappear from the historical record in the late 17th century, starting in the upper classes of Northern Europe, and then gradually filtering down through society over the next 200 years. By the early 20th century, the concept had vanished almost entirely. And what changed? Artificial light. Street lighting came first. Paris began installing oil lamps in the 1660s. London followed. And then domestic lighting improved. And suddenly, the night wasn't this impenetrable darkness that drove you to bed at 8 or 9 o'clock. You could stay up. You could socialize, work, read. The evening expanded. Bedtime got pushed later, and that middle of the night waking period got squeezed out. The two sleeps merged into one. Now you might be thinking, okay, that's interesting history. But is there any biological evidence for this? And the answer is yes. And it comes from a beautifully simple experiment. In the early 1990s, a psychiatrist named Thomas Weir at the National Institute of Mental Health took a group of healthy volunteers and put them in conditions that mimicked a pre-industrial light environment. 14 hours of darkness every night. No screens, no electric lights, no books. Just darkness. And he kept them in this regime for several weeks. At first, the subjects slept enormous amounts, probably catching up on accumulated debt. But once they'd stabilized, something striking emerged. They settled into a pattern of, you guessed it, 2 distinct sleep periods, separated by 1 to 2 hours of quiet wakefulness. Their sleep architecture during this wakeful interval was interesting too. They weren't fully alert. Their brain activity showed elevated levels of prolactin, a hormone associated with a calm, meditative state, something like the brain's version of a peaceful twilight. It was a mode of consciousness that modern people essentially never experience. Wehr's experiment suggested that biphasic sleep isn't just a cultural artifact. It might be a latent biological program that's still in us, masked by our artificially extended days. And that raises a genuinely provocative question. When someone wakes up at 2 or 3 in the morning and lies there for an hour before falling back asleep, are they experiencing insomnia? Or are they experiencing a perfectly natural sleep pattern that we've pathologized because it doesn't fit the industrial schedule? Now, I want to be careful here, because this is one of those areas where a fascinating idea can get oversimplified. Not everyone who wakes in the middle of the night is having a blissful, segmented sleep experience. Genuine insomnia, the kind that comes with anxiety, racing thoughts, daytime impairment, is real, and it's distressing, and it needs treatment. The point isn't that all middle-of-the-night waking is fine. The point is that our definition of normal sleep is narrower than it probably should be, and that some people who are sleeping perfectly well for their biology are being told they have a disorder because their pattern doesn't match the idealized 8-hour block. And this connects to something broader about how sleep varies across cultures even today. If you look at sleep patterns in pre-industrial or semi-industrial societies, and several research teams have done exactly this, you find a lot of diversity. Jerome Siegel at UCLA led a study published in 2015 that tracked sleep in 3 hunter-gatherer groups: the Hadza in Tanzania, the San in Namibia, and the Tsimane in Bolivia. These are populations living without electricity, without alarm clocks, without the whole apparatus of modern sleep hygiene advice. And what Siegel's team found surprised a lot of people. These groups slept, on average, about 6.5 hours a night. Not 8, not 9, 6.5. They didn't nap much. They didn't go to sleep at sunset. They typically stayed up 3 or 4 hours past dark, socializing around fires. And they woke up before dawn, often in the coldest part of the night, which suggested that temperature might be as important a cue as light in regulating their sleep timing. This finding complicated the narrative that modern society is chronically sleep-deprived compared to our ancestors. Maybe our ancestors weren't sleeping 9 or 10 hours either. Maybe the sweet spot is more variable than we thought. But, and this is important, those hunter-gatherer groups also weren't dealing with artificial light after dark, shift work, social media at midnight, or any of the other factors that genuinely do disrupt sleep in industrial societies. Their 6.5 hours might have been 6.5 hours of very high-quality, well-timed sleep, which is a different thing from 6.5 hours grabbed in fragments around a chaotic modern schedule. So what does all this tell us about what good sleep actually means? It tells us that the answer is more personal, more culturally shaped, and more biologically variable than any single recommendation can capture. The 8-hour rule is a reasonable average, a useful guideline, but it's not a law of nature. Some people genuinely need 9 hours. Some thrive on 6.5. Your chronotype matters. Your age matters enormously. Think about how we discussed adolescent sleep shifting later, and consider that older adults tend to shift earlier again and often sleep in shorter bouts. Your genetics matter, as the Patacek and Fu work on clock gene mutations showed us. Even your season and latitude might matter, since some research suggests people naturally sleep a bit longer in winter darkness than in summer light, which makes intuitive sense if you think about it. The real danger isn't sleeping 7 hours instead of 8. The real danger is the things that erode sleep quality regardless of duration. And in the modern world, those things are everywhere. Let's talk about a few of them, because they connect directly to the mechanisms we explored earlier. Light is the big one. Remember the suprachiasmatic nucleus, that tiny cluster of cells that serves as your master clock? It takes its primary cue from light hitting specialized cells in your retina, the intrinsically photosensitive retinal ganglion cells, which contain a photopigment called melanopsin. These cells are particularly sensitive to short wavelength light, the blue end of the spectrum. And what do modern screens emit in abundance? Blue light. When you scroll through your phone at 11 at night, you're sending a signal directly to your SCN that says, essentially, it's still daytime. Your melatonin release gets suppressed. Your circadian phase gets pushed later. And the insidious thing is that it doesn't feel dramatic. You don't feel a switch flip. You just find yourself not quite sleepy yet, staying up another 20 minutes, another half hour, and the debt accumulates so gradually that you barely notice. Charles Czeisler at Harvard has been studying this for years, and his lab has shown that even a few hours of evening screen exposure can shift your circadian clock by an hour or more and suppress melatonin production by over 50%. A study from his group published in 2014 compared people reading on an iPad before bed to people reading a printed book. The iPad readers took longer to fall asleep, had less REM sleep, produced less melatonin, and felt sleepier the next morning, even after the same amount of time in bed. The effect was measurable, reproducible, and dose-dependent. More light exposure meant more disruption. And light is just one factor. Temperature is another, and it's one that sleep researchers wish more people knew about. Your body needs to drop its core temperature by about 1 to 1.5 degrees Fahrenheit to initiate and maintain good sleep. This is part of the circadian program. Your temperature naturally dips in the evening and reaches its lowest point in the early morning hours, roughly 2 hours before your typical wake time. Anything that interferes with this cooling, a room that's too warm, too many blankets, heavy exercise right before bed, can fragment your sleep even if you don't fully wake up. The ideal bedroom temperature for most people is somewhere around 65 to 68 degrees Fahrenheit, which feels cool, maybe even a little cold. But that's the point. Your body wants to shed heat. There's a lovely bit of engineering in how your body manages this actually. When it's time to sleep, the blood vessels in your hands and feet dilate. They open up wide, bringing warm blood to the surface, where the heat can radiate away. That's why your extremities often feel warm right before you get sleepy. It's not that your body is heating up. It's that your body is dumping heat through your skin, lowering your core temperature. It's a radiator system, if you think about it. And it works best when the ambient temperature is cool enough to actually carry that heat away. So you've got light, temperature, and then there's the chemical environment, which brings us back to something we talked about much earlier, adenosine and caffeine. But there's a dimension to caffeine that most people don't fully appreciate, and it has to do with its half-life. Caffeine's half-life in the average adult is about 5 to 7 hours. That means if you have a cup of coffee at 2 in the afternoon, containing roughly 200 milligrams of caffeine, by 9 or 10 at night, you still have about 100 milligrams circulating in your system. That's roughly the equivalent of a full cup of coffee, sitting in your brain, blocking adenosine receptors at the exact time your body is trying to build up enough sleep pressure to push you into deep non-REM sleep. And you might fall asleep fine, Caffeine doesn't always prevent sleep onset, but it can reduce the amount of deep, slow-wave sleep you get, which is exactly the sleep that's most important for memory consolidation, for glymphatic clearance, for all the restorative processes we talked about. You wake up feeling unrefreshed and reach for more coffee, and the cycle deepens. You drink more coffee the next morning because you're tired, which pushes more caffeine into the afternoon, which steals more deep sleep the following night, which makes you more tired the next day. And around it goes. It's a self-reinforcing loop, and millions of people are caught in it without ever realizing that the thing they're using to fix their tiredness is one of the things perpetuating it. And look, I'm not here to tell you to quit coffee. That's your call. But understanding the mechanism at least lets you make an informed choice. If you shift your last cup to before noon, you give your body a much better shot at clearing most of that caffeine before bedtime. It's not a perfect solution. Everyone metabolizes caffeine a little differently, and genetics play a role there too, which we touched on when we talked about those clock gene mutations. But for most people, a noon cutoff makes a meaningful difference. So here you are, lying in the dark, hopefully in a cool room, hopefully without too much caffeine still circulating, and your body is doing all of these things we've been talking about. Your suprachiasmatic nucleus has been tracking the fading light all evening, telling your pineal gland to ramp up melatonin. Your adenosine levels have been building all day, pressing on the sleep side of that flip-flop switch we described. Your core temperature is dropping, heat radiating out through your hands and feet. The wake-promoting neurons, the ones firing histamine and norepinephrine and orexin, are gradually losing their battle against the sleep-promoting neurons in the ventrolateral preoptic area. And at some point, the switch flips. Not gradually, but all at once, the way Clifford Saper described it. You cross a threshold, and you're asleep. And this is where I want to bring things together, because we've covered an enormous amount of ground tonight, and it might be nice to see how it all fits into a single picture. Not as a review exactly, but more like stepping back from a painting you've been looking at up close, section by section, and finally seeing the whole thing at once. Here's what we know, as of right now, in the early 21st century. Sleep is not one thing. It's a sequence of distinct physiological states, each with its own brain activity patterns, its own neurochemistry, its own biological purpose. Non-REM sleep, especially the deep, slow-wave stages, is when your brain replays and consolidates memories, when those slow oscillations and sleep spindles and hippocampal ripples work together in that beautifully timed coordination that Jan Bourne's lab spent years unraveling. It's when the glymphatic system opens up, cerebrospinal fluid flushing through the interstitial spaces, carrying away metabolic waste including beta amyloid, the protein implicated in Alzheimer's disease. It's when growth hormone surges, when tissue repair accelerates, when your immune system does some of its most critical work. REM sleep, meanwhile, is when your brain processes emotional memories, stripping away the sharp edges of difficult experiences, integrating new information into your existing understanding of the world, making creative connections between ideas that seemed unrelated during waking hours. That Wagner and Bourne study, where people were more likely to discover the hidden shortcut after a night of sleep, that's REM doing its thing, shuffling and recombining, finding patterns in the noise. And all of this is orchestrated by two overlapping systems. Process C, the circadian clock, tells your body what time it is, syncing your internal rhythms to the external world through light and temperature and social cues. Process S, the homeostatic drive, tracks how long you've been awake through the accumulation of adenosine and tells your body how much it needs sleep. When those two processes align, when the circadian clock says it's nighttime and the homeostatic pressure says you've been awake long enough, sleep comes naturally. When they're misaligned, because of jet lag, shift work, social jet lag from mismatched schedules, blue light exposure pushing your clock later, or caffeine masking your adenosine signal, that's when things start to go wrong. Now here's what I find genuinely exciting about where sleep science is headed. For most of the 20th century, sleep was treated as a passive state, something that just happened when you stopped being awake. The brain shuts down, you're unconscious for a while, you wake up. That view is completely, utterly wrong, and we now know it. Sleep is one of the most active, complex, and purposeful things your brain does. But even with everything we've learned, from Aserinsky's flickering eye movements in that basement lab in 1953, through the discovery of the flip-flop switch, through the glymphatic system and the two-process model and all the rest, we still don't have a unified theory of why sleep exists. We have a collection of important functions, each well supported by evidence, but no single overarching explanation that ties them all together. And some researchers think that's because we've been asking the wrong question. Instead of asking, why do we sleep? Maybe the better question is, why would any organism ever be awake? Because sleep, or something very much like it, appears in every animal species ever studied. Even organisms without brains show rest states that share features with sleep. Wakefulness is metabolically expensive, dangerous. You're exposed to predators, you're burning energy, you're accumulating cellular damage. From an evolutionary perspective, the default state might actually be something closer to sleep, and wakefulness is the special state that evolved because it was useful for finding food, avoiding threats, reproducing. That's a provocative idea, and it's not universally accepted, but it reframes the question in a way that I think is really interesting. It suggests that sleep isn't a problem to be explained. It's the baseline. Consciousness is the thing that needs explaining. There's also a growing understanding that sleep isn't just important for the brain. We talked about the immune system, about metabolic regulation, about the cardiovascular data from the daylight saving time studies. But the picture keeps getting richer. Research published in the last several years has shown connections between sleep disruption and gut microbiome composition, between sleep and wound healing rates, between sleep and the effectiveness of vaccines. One study found that people who slept fewer than 6 hours the night before receiving a flu vaccine produced less than half the antibody response of people who slept normally. Half. Your immune system literally learns less effectively when you're sleep deprived, which makes sense when you think about what we know about memory consolidation. The immune system has its own form of memory, and it seems to benefit from sleep in ways that parallel how the hippocampus benefits. And then there's the frontier that connects back to Mike and Nedergaard's glymphatic work, the relationship between sleep and neurodegeneration. We touched on this, the bidirectional cycle where poor sleep increases amyloid accumulation, and amyloid accumulation disrupts sleep. But what's emerging now is even more nuanced. Researchers are finding that it's not just the total amount of sleep that matters, but the specific architecture, the proportion of deep, slow-wave sleep, the timing and coupling of those oscillations and spindles. People in the early stages of Alzheimer's often show disrupted sleep architecture years before they show cognitive symptoms, which raises an extraordinary possibility that sleep monitoring could become an early diagnostic tool, not a cure, not a treatment, but a way to catch the disease before it causes irreversible damage. There are labs working on this right now, developing algorithms that can detect subtle changes in sleep EEG patterns that might signal the very earliest stages of neurodegeneration. It's still early days, but the potential is remarkable. And I think what strikes me most about all of this, the thing that makes sleep science feel different from a lot of other fields, is how personal it is. Every single person listening to this has a direct, intimate, nightly relationship with the subject. You don't need a telescope or a particle accelerator or a deep-sea submersible. The laboratory is your own bed, your own brain, your own body cycling through these ancient rhythms that connect you to every sleeping creature that has ever lived. When you feel yourself getting drowsy, that's adenosine pressing on Saper's flip-flop switch. When you dream, that's your brainstem activating the same circuits that Osirinsky stumbled onto 70 years ago. When you wake up and the solution to yesterday's problem suddenly seems obvious, that's the same process Lowy experienced when he dreamed of the experiment that proved chemical neurotransmission. You are, right now, doing something that science still doesn't fully understand, and that's kind of wonderful. There's a quote I think about sometimes from Alan Rechtschaffen, who was one of the pioneers of sleep research at the University of Chicago. He said, "If sleep does not serve an absolutely vital function, then it is the biggest mistake the evolutionary process has ever made." And he said that in 1971, when we knew so much less than we do now. Every decade since then has only strengthened his point. Sleep isn't optional. It isn't wasted time. It isn't a design flaw in the human operating system. It's the maintenance cycle, the consolidation phase, the cleanup crew, the emotional therapist, the immune trainer, the creative incubator, all running simultaneously in a beautifully orchestrated sequence that repeats 4 or 5 times every single night of your life. And the fact that you're here, listening to someone talk about it, already feeling the pull of it, that's your biology working exactly as it should. The adenosine is there, the melatonin is rising, your core temperature has dropped, the world outside is quiet, and your brain is ready to do what it does best when you let it. So let it. Let your thoughts soften. Let the details we talked about tonight, the flip-flop switches and the sleep spindles and the glymphatic channels and the circadian rhythms, let them blur together the way things do when you're drifting off. You don't need to remember any of it right now. Your brain will sort through what it wants to keep. That's literally what it's about to do. The slow oscillations will come. The spindles will fire. The hippocampus will replay whatever it found interesting. And by morning, some of this will have settled into something that feels like understanding. For now, though, there's nothing you need to do. Nowhere you need to be. No problem you need to solve. Just this, the quiet, the dark, the slow unwinding of a mind that has been awake long enough. Your body knows how to do this. It's been doing it every night of your life, and every night of every life before yours, stretching back through millions of years of evolution to a time before there were even brains to sleep with. Whatever sleep is, exactly, and we're still figuring that out. It's yours, and it's here, and it's waiting. So let it take you. Good night.